Always forget to turn the microphone on at first. What's up, Rosa? I should just go get my book on protozoa right now. Not sleeping all night has its perks. Oh yeah. Is it too hot to sleep where you are or something? Why aren't you sleeping? Because I feel like not sleeping all night also has some minuses that don't involve me. I'm not sure which one this is. I don't know that I've ever seen anything quite like this before. It looks like a little tiny vase attached to the plant. I'm sure it's a ciliate. Insomnia, oh okay. Yes, Peng, it's latched onto the plant stem there. It's not actually a plant stem, it's actually algal material, but it's latched onto it. Who needs sleep? Well, I mean, occasionally I need sleep. It looks like Spirocona or something in that group. Kind of like a little tiny vase attached. Not totally sure. It says here that Spirocona is mostly attached to zooplankton, though. This is definitely attached to algae. So, question mark as to what it is. Doesn't look like it is feeding. Yeah, it may not be. It may just be chilling. Although there's a lot going on inside here. Like this is pretty busy. For something that's not feeding. Also, that skill bar is wrong. We're at 20 times 3. We're at this size. There you go. Hey, Killathon, how you doing? I don't think it's an alien, but you never know. These little guys over here are diatoms. Right here. And this is Cladophora, which is a group of green algae. Could be digesting, yeah. It seems pretty active, but I don't see anything going into it. So, not sure. Oh, somebody stuck their head in my... Oh, it's a little Rotifer. There's a bunch of cool stuff in here. So this is from the pond, my backyard pond. Just checking in on it to see what's growing. And what we can find in it. It looks like there's a giant worm here. I mean, it's a small worm, but relative to everything else we're seeing, it's giant. Clostridium. Maybe. I don't know. I didn't find it in my book, which is the only resource I have. That there's definitely a worm, though. We've seen their kind before. I think the last time we had one of these on here, it was... Oh, look at you, putting a name on it. Botulinum. I'll look it up. Let's see how close we are. Clostridium. Clostridium. Botulinum. This little thing? don't know. See the inner working parts of the worm here. Pretty cool. You hate not knowing. Well, we need to have somebody who's an expert in other things that aren't diatoms here. They probably could tell us. That looks like a nematode worm. This is how they usually act. Just little spazzes. And there's Cenodesmus and diatoms all over the place right there. So those are all diatoms. Those are Gumphonema. I think I looked these up before because there's just basically a monoculture of this diatom in my pond. And I think they are Gumphonema johnsoni. I think that's what I decided they were. There they are with a little bit less contrast and the polarization's not so strong on them. That's a nematode sticking its face in there. Yeah, definitely little clusters of diatoms. You can see there's also, that's the Cenodesmus right there. So its little spines on the ends have a bit of gas in them that helps them float. And then it looks like there's a rotifer right there that's kind of making everything vibrate. It's not them, it's that guy right. Oh, now there's a nematode worm butting in. This guy right here is a rotifer. And then that was a nematode that knocked everything around. It's like a universe of things that will give you diarrhea. Well, they could. Potentially give you diarrhea, I suppose. Most of them probably don't have any effect on you at all. Most of these guys are probably just, they're going to get into the acid of your stomach and then just die instantly. But I don't recommend eating them. I heard on Pacific Plankton Stream, though, that they taste like spam. So, you've never seen a Cenodesmus that short? Well, they come in a 4-chain variety, like these guys, and an 8-chain variety, and a 2-chain variety, and a 1-chain variety. So they have a whole bunch of different sort of colonial phases. And at least some of the research suggests that the number in the colony is a response to fish eating Cenodesmus. So when they're broken open, never odd numbers. It's always duplicates of 2, except for when there's 1. I guess that's an odd number. When there's fish, thanks Nematode. When there's fish in the water that eat them, it releases a chemical and it lets them know that it's time to start making longer and longer colonies. And so if it's water that's collected from like water with fish in it, it usually has 4 or 8. Yeah, I got yeeted. Nematodes are kind of interesting. I'm always surprised at how strong they are. I mean, they smack everything around pretty good in there. How's it going, studio? We're looking at some stuff in the light microscope tonight. It's a change of pace. Normally I do a little SEM stream in the afternoon, but I had some other work I had to get done, also on the SEM. I just didn't feel like sharing it, because it was maybe less interesting for people. It was just me taking pictures of the same 2 diatoms over and over again, but it had to be done. So we have a paper that's coming out on June Lake, which is one of the lakes that I study, a paleo record of June Lake. And we've been working on it for many years now. Not the paper, just the research. And they wanted to make like a graphical abstract using some SEM images for it, because now that it's accepted, we have to make some, gussy some things up and potentially make something to put on the cover. So I went into the SEM lab and just collected some of my some good pictures of the species that we were looking at. So, you'd like to read it when it comes out? Well, I don't know when it'll be released. It's embargoed right now, or I'm told it's embargoed. You're going to June Lake next week? Oh. There's a diatom in June Lake that I described. And it's one of the two that are in this project. And then there's another diatom in there that I probably need to describe, and at least two others that I've identified as probably need to be described, but I just haven't gotten around to them because they're lower priority or problem diatoms. So they weren't as easy, basically. So there was some sort of low-hanging fruit in there, and I took it first. And then my graduate student Laura just got back from June Lake and Gull Lake, because she's doing research in Gull Lake. And that's a rotifer, that's a deltoid rotifer. She's spinning the water around using her corona, which is the little cilia at the top. And it'll give us a nice look if we zoom in here on this. You can see the mastax, which is sort of their jaws eating here, and this tornado that she's making by spinning the cilia at the top of her head. And this little feature right here is the antenna. It's like a sensor tool that they use, and its mouth is usually nested in here somewhere. And then the stuff that's spinning, some of it goes into its mouth, into the mastax, and then that's its digestion right there that you're seeing in action. And because we're super zoomed in on it, I can only get so much of it in focus at the same time. But that is definitely a deltoid rotifer. You can see the gyre she's creating right here. This constant whirl. It's a little tiny guy that came zooming in right there. That's a ciliate as well. So, and then this is Cladophora, which is all over in my koi pond on the sides. The goldfish eat this, and they probably also are scraping some of these diatoms into their mouths as well, and anything else that's on it. So, I'm a problem diatom. It's true. It doesn't, it's not a simple species. It's a complicated, the other primary diatom in there is a complicated group. And it's not that I am afraid of going after complicated species. It's that it required me to send it off to somebody who's an expert in that group. And they took a look at it, and they just haven't really done much with it. So it's been sitting because they let it sit. But we think it's a new species. And it's just a challenge because of where it is. The type of diatom that it is makes it difficult. Whereas the other one was just a Stephanodiscus, which is pretty easy by comparison. But it had some of its own problems. Oh, look. It's a heliozoan. Just sitting there, hanging out. Boop. You see all the spines radiating out from it. Those are like little tiny skinny pseudopods. It's little tiny legs they have sticking out in every direction, basically. Hello, Sanders. Yeah, it's a pretty one, isn't it? It looks very stellar in this. I'll make this look more like night sky. Here we go. With the bubbles floating by of probably bacteria bubbles. Doing an okay job of not making my desk vibrate too much. They were once called sun animalcules. The animalcule name is pretty funny to me in general, but yes. They do look a little bit like suns. And then you can see all the diatoms, they're just covering all this Cladophora, like all the way around. All those little Gumphonemas. And I'm not sure what else is in here. I'll just tool around a little and see. So I'm on the 20x objective. And then that little box is where I zoom into. I guess I could get rid of that for you. But you can see it's just like a nest of benthic diatoms attached to these little stalks to the Cladophora. So, and when I don't feed my goldfish, this is what they eat. They just come over and chow on the sides of the koi pond. And when I went out today to collect some samples, actually I went out just before I started streaming. I just scraped some of it in the darkness. But I was kind of surprised at how little was coming up, so they must be getting kind of hungry. In the summer they're probably a lot more active. That's another rotifer right there. Deltoid rotifer. It doesn't look like it's doing very well. Over towards the edge of the slide. Sometimes we find some cool stuff just digging around in these samples. I started just before we started streaming, I just sort of scanned through them and I saw some gastrotrix. So I know there's some in here. I don't know that we'll find them again because there weren't that many of them. But they're very cute. Their name means hairy belly, hairy stomach. And they look like little tiny slugs with hair sticking out in every direction. So if we find one of those, I might chase it around a little. Some little spinning ciliate down there. There's another big nematode worm. Thinking the filamentous are pseudoparenchymatus. You're not a great identifier. Well, I don't recognize that name, so you're doing better than me. If it's not a diatom, I probably don't know what it is to species. Oops, hello. Why are you moving around? Oh, nematode. Oh, that's cool. Look at that guy. We could keep him on the field of view. I think he's still wiggling. Just leave it alone, nematode. Yeah, he's definitely wiggling. His little cilia are like in a ring around the outside. It's otherwise a globe. Slither IO. I think my daughter plays that game. It's just like encircle things to collect points or something. Knock out other people. Kind of like worms. That's a cool little guy. Here comes the Nematode. Yep. They just can't help themselves but screw everything up. There goes our subject. Now it's playing catch with it. The worms eat the other worms. Okay. You spent many hours playing worms. Yeah. Well, let's see if I can get this worm to leave my ciliate alone so we can look at it a little more. Pretty cool. Seems kind of sleepy, but it's definitely alive. Yeah, it's moving. The little cilia are moving on their own down here, like on this fringe that goes around here. It's definitely moving. It's not whirling around like you usually see them. I think if you saw these things, if we could somehow get them in the SEM without them turning into like something that looks like a deflated bag, they'd probably be terrifying to you. Some sort of beholder. Crazy monster. There, now it's kind of moving. I think its mouth is here in the middle of all these little cilia. And it actually looks like it has a diatom inside it. I don't know if you can see that. When it turned a little bit, I saw right here, I can see this little boat shape that looks exactly like a diatom. And I'm pretty sure it's digesting a diatom as we watch it. It's not visible right now. Oh, it is slightly visible right in here, I think. Like this. I think that shape is right there. You can see it. I think that's a diatom that it's eating. It's digesting. These little things that we see out here are on the margins up here. They're shaped like little submarines. I think it's got one in its belly. That's probably why it's not moving very much. It's satisfied. It's just hanging out. Pretty cool. Some of those little ciliates, I think that one is Strombidium or something like that. That's like perfect, really round one with a little fringe on it. But some of them are actually like oxygen is toxic to them. I think they're called Loxo something or other. I don't remember the names of all of them very well. It's taking a food nap, yeah. It's got a little torpor going on. And these ones are like Epistylus. Connected on these chains. And then you can see they also have the same kind of little fringe of cilia that run around the outside of the top. And they're also creating a little current. And you can see this one here is doing it too. Like you can really see the cilia in here whirling around. And they're living together in like a little colony, like flowers. But you can see sometimes when they get hit in the face by things that are being sucked towards their face, that they'll retract on these little stalks that they're on. They can kind of pull themselves back down towards the surface they're attached to. So like in here you can see it's actually attached, and they can yank themselves back in to be safe, basically, or to try to get safe, I guess. Not necessarily safe. Here comes the nematode again. How do microplastics affect these organisms? Well, most people don't realize that there's a whole nother class of microplastics called ultra microplastics, which are basically everywhere now. And they're just in the air. They find them in mosquitoes. They find them in everything. And, the sort of influence that microplastics probably have on these organisms is probably pretty small, but the normal effect of microplastics on organisms is that they don't digest. So if they swallow it, it just ends up being stashed in their stomachs. And then that means they don't have any, that takes up space that food would normally go through. Some of the organisms, they'll just pass through them relatively harmlessly. So Studio says that they found microplastics in all of their samples, even obscure samples out in the Mojave Desert. Yeah, you don't have to be near people. They're blown around in the air. They're being transmitted by basically everything. And we did some studies on fish from the Wabash River, where we looked at the gut content of fish that were collected in the 60s, 70s, 80s, 90s, all the way through on the Wabash River. And we found microplastics in the fish in the 60s, starting mostly with fibers, because we were doing a lot of plastic microfibers in like synthetics. And they're probably just being consumed incidentally by fish, but the normal consequence for microplastics is for organisms that have a digestive tract that they can't escape from. They end up just getting stuck in them, and then they pile up and die. But they also, if that organism gets eaten by something, it just gets transmitted up the food web. So the same kind of process happens regardless of what level you're looking at the organisms, because the plastics that get stuck in this, anything that grazes on it gets stuck in them, until it reaches some sort of terminal position in the bodies of something. There's a little diatom out here trying to fight the current. That's just the consequence of having something that's not digestible, that's small enough to fit into their mouths. Some of the moderate-sized ones, because these things you can see they're filter feeding, or they're feeding by sitting still, and it looks like the cilia are spinning, but they're just doing this. They're waving them, like we saw the other one, the round guy, doing. They just wave them, and they're creating a current by waving them around continuously, and then whatever comes towards their mouths, basically, they consume. Some of the ciliates and some of the larger nematodes out here punching these guys. Sometimes they will reject things that they eat, so they can spit it out if they know it's not food. And especially the larger pieces of plastics, they probably can spit out. The very small pieces of plastic, the ultra micro plastics, there's probably no option other than to consume them, because they don't know that they're eating. Not if you can imagine. It's so small. So RCPD33, thank you for the follow. I don't know what that face is for, exit, but You don't want to eat plastic? Is that what you're trying to tell me? Because you also eat plastic. Whether you know it or not, it's accumulating in your body as well. And they did a study where they looked at people and they checked out their poop as well. And in 9 out of 9 people, regardless of where they lived, they found plastics in their poop. So it's in everything. There's just no two ways about it. It's, you know, if you eat fish is eating plastic, and you eat the fish, then you're eating the plastic too. You know, some things we can avoid because we don't eat the digestive tracts or whatever. But dumb question. But can diatoms digest a little since their shell is made of silica? Diatoms don't typically eat. They are photosynthetic, like most algae. Almost all of the diatoms make their own food from sunlight and carbon dioxide. And but I should also point out that there are examples of diatoms that are heterotrophic. I think they mostly eat detritus, although I've never really looked into the literature on it because it's something that's so foreign to me. But I do think that there's evidence that some diatoms can switch to eating, but they don't really have mouths, so they just I think they just absorb nutrients rather than like sunlight, and they get it mostly from, I think it's mostly from detritus, so like pre-mineralized or partially mineralized material. D-Lurks, hello. Vegetarians are not safe either because it's also in the soil and water that feeds the plants, yeah, and also anything that's in the air settles on to plants. There's no avoiding plastics, so it's a pretty critical problem. You know, for humans, plastics are relatively harmless, at least the plastic itself is. One of the problems with plastics of all varieties is that they are surfaces that things can absorb onto, and that means that you could potentially be eating something that is a persistent pollutant in the environment that's absorbed onto the plastics themselves, and we don't really know what the effects of those are. Look at that colony of diatoms. That's just ridiculous how many diatoms are in just this little area here. There's hundreds or thousands of them right just in this little tiny bit right there. It's a pretty big colony, but every piece of Cladophora that we've looked at here has had just like massive colonies of diatoms growing, and it's all the same species basically. We put some of this stuff in the SEM, and if you're here right at the beginning of this, I mentioned that I think it's Gomphanema johnsoni. So I don't think Pacific is here, but Gomphanema. There you go. And if you click on that link and click on johnsoni, you'll probably actually see the diatom that we're looking at. I could look it up for you, or you could do that. There's another nematode just butting in like they do. They're like, oh, are you trying to stream? Do you mind if I get in? But it was basically just a monoculture of this like one diatom. There are some other diatoms that are in here. They're very small. This johnsoni is like, I think it's something like 20 to 40 microns. They're pretty small, but everything else that I saw is like really small, in like 2 micron range. So some of these little things that are just little bumps on the plant are actually diatoms. They're just so small that we can't see them even at 40x. We would have to take it up to 1000x to really see them well. But in the SEM, you can see them easily, so it doesn't matter how small they get. The smallest diatom I've ever seen in the scanning electron microscope was about... Just what happened? Oh, Evil Ozzy's hosting. Oh, did you raid? Oh, thank you, Evil Ozzy. Look, I did something here. Let's see. Evo. There you go. If you're not following Evil Ozzy, you should go give him a follow. And yeah, you've got your own command now. Pond microbes, yeah, that's what we're looking at, as usual. For evening streams, I'm either looking at the moon, or like with my camera, or we're doing a little bit on the light microscope, looking around. These are samples from the pond that is behind my house, a koi pond. It's very small. It just has a bunch of goldfish in it. It doesn't actually have any koi, because I think the raccoons all came in, or maybe it was a hungry crane or something. Hungry heron came and ate all of my koi. They left some of the goldfish, or most of the goldfish behind, but that happened like a year and a half ago or something. All the koi just disappeared from it one day. You know, I didn't see them anymore. I don't know where they went. Yeah, raccoons are always a problem like that. Space but in a lake. You love my microscope a lot. I also love my microscope. I can't quite see the name for it, but it's right down here. I can't get it up with there, right there. It's a Leica Dm 2500. It's actually my lab microscope, but I have two more in my lab, and I have this one at home because my wife and I are both diatomists, and through the corona outbreak, we've been working from home. Here you can see one of those little heliozoans, and you can see it's got a really long little thread right here, one really long one. It's pointed out in the direction. It's kind of traveling in that direction too. Super slow, but you can see there's another little long thread, and there's another little long thread. They have these long sort of skinny pseudopods that stick out. It's an amoeboid. And then, you know, there's like a central body right there that it's living in. You can actually see it's got threads sticking out, these little pseudopods threads sticking out in almost every direction. So there's a lot of sort of amoeboids in pond samples, and when they're shaped like little suns with like a central area that's rounded, and then a whole bunch of radiating little tiny skinny things coming off of them, usually they're just lumped into with the heliozoans. So helio means sun. Major scope jealousy, yeah. What is the bug on the screen? Oh, are you talking about this thing? I think it's a heliozoan. I think I answered that question. The name of the scope is a Leica. Leica is a major brand of microscopes. They also make cameras and some other things. And then the model number is DM 2500. So if you want to buy one, this model with, it has a 5, a 10, a 20, a 40, and a 100x objective. And they are N-plan, which is like really nice for differential interference contrast objectives. Except for I don't think that the 5x is an N-plan. I don't think the 10x is either. But it's a special microscope because we're looking at stuff in differential interference contrast lighting. And that's what makes the microscope cost so much. You don't need differential interference contrast to see things, but it provides this sort of three-dimensional looking high relief sort of structure to most of the images that I collect from it. And for diatom work, it's pretty critical to have DIC. So the differential interference contrast is like standard operating procedure for diatom images. It's because we can adjust the contrast however we need to in order to see little features. And then, yeah. And then it's got a really nice camera on the top. That's not one that you can typically buy with the microscope. That's an actual real camera up there, which is my Olympus OMD EM1 Mark II body. I also have a Mark V and an EM5 Mark II body, sorry, and a EM1 Mark I, EM1 Mark III. Sorry, I've got like 3 different camera bodies now. My niece gave me back the one that she had borrowed, and I was thinking I might actually switch over to streaming with that as my camera instead of just using a webcam. They look really nice, and I've got a really like cool lens that she was using on it that my sister lent me, and I haven't even tried it yet, which my sister will be very disappointed in me if I don't at least try to stream with my other one. But plugging 2 cameras into my computer seems like 2 big cameras seems like overkill to me. So we'll see. Maybe I will, maybe I won't, but it'll be fun to give it a try at least once, to use it as my webcam. That's kind of ridiculous to have a $1,000 webcam, but especially because my webcam image is so small usually. There's a little nematode doing their sneaky wiggly thing. This one's not going very far, so maybe we can take a look at it. It's pretty cool. Yeah, not bad. Evo. What do mosquito larvae look like up close? Well, you can find lots of pictures of mosquito larvae. They have a siphonal tube that comes off the back end of them, and other than that, they just kind of look like a little stick, but with legs. But I've never actually seen a mosquito larva in my koi pond. They're mostly they can mobilize kind of a lot, and probably they get eaten by my goldfish kind of a lot. So did you look up the price for it? Yeah, this came from a research. This is my research grade microscope, and the other 2 also were bought. One was bought up with startup money, and one was bought. These 2 were bought with grant and funding from my department. My college. And I tried to design this one to be a little bit different because the ones that I have at the lab all have built-in microscopes, built-in camera and software for measuring things and sort of documenting things a little bit better. And we bought this one after I started streaming, so I kind of wanted one that was a little bit more geared towards streaming and just doing work on the microscope from home. Eventually it'll go back to the lab, though. It's not my personal microscope. It's just that I'm not I was trying to keep people's germs from being on it because your eyes are constantly on the eyepieces. And so I thought it was a really bad idea to have a microscope where there was a bunch of people who I don't know what they do during their days, putting their eyes on my microscope and then having to constantly clean it. So I just brought it home so that my wife and I could work here. And then, and not everybody in my lab is vaccinated yet still, even though it's been a while. So there's that. Like, I think if everybody was vaccinated, I probably would be more inclined to leave it at school. But my daughter is not old enough for vaccination yet, and so until that happens, we also probably won't be bringing it back and forth to school. So, gotta wait. If people in my lab really wanted to use it, they would get vaccinated, and then we wouldn't have this question. That is another deltoid rotifer. It's looking kind of fuzzy. I don't know if it's covered with something. It's looking pretty fuzzy. Yeah, pink eye. I don't know about pink eye, but once you get it, once you get like something on the microscope, yeah, you have to constantly clean them. And my lab tech was responsible for cleaning the ones in the lab. And hello. And you know, so that's okay. Oh, this one is actually feeding. We saw one of these right at the beginning of the stream. And it was closed in, it wasn't feeding, but that one has got its little lid open, and you can see it's got its cilia out. And it looks like it's feeding. It's flapping away. The other one we saw was just kind of like digesting. This one I looked up, and I thought we had it. I thought I had it pretty well sorted out, because I thought it looked like something in my little booksy here. It's pretty close to this thing they're calling Spiracona. And that's what it looks like to me. It has like a little trap at the top, and it's kind of vase-shaped. It says they're usually found on zooplankton. But it says one of these cells has been, oh, this one says it's been eating blue-green algae, so you can see that. Yeah, so I'm not sure if that's what it is, but it's the thing that looks closest to it in the book here. Super cool. It's got like a little trap door at the top. Can you see that? It's like this little thing. I think when it's feeding, that's open, and then when it stops feeding, I think that clamps down and all the little cilia come inside the body. It's pretty cool. See, it's got some stuff in its belly. It's got little stuff sticking out the top part. Life is crazy. Some living things, the little tiny living things. So that's a diatom right there. And this thing is just slightly bigger than these guys. And I think the average size on these was something like 40 microns. So that thing's probably like 70 microns long. It's not very big. We can only see it because of the, you know, I'm zoomed in and we're on the 20x objective. You know, it's, I think I could take it up to 40. Let's see if we can do that, how it looks. Oh, I startled it. Yeah, you see it's closed now? It's closed its little thing, and now it's going to open it for us, hopefully. There it goes. Popped right back out. Okay, let's see if I can fix that view. There you go. So this is what it looks like at 400 times magnified times 3. So we're super zoomed in on it. And let me see if I can fix the lighting a little. Just gonna tweak some settings. And then I'm gonna just gonna try to move it up a little so we have the whole thing in the field of view for you. Look at that little guy, isn't he pretty? Now you can really see all the stuff on the inside. And then there's this little sort of trapdoor structure here at the top that we saw pop open. And all this stuff folded out from inside of the body. I think this little cavity right here is where it was being stored, and it just flipped out, and then all the little hairs are wiggling. And you can see that the distortion on the water, and you can see it pulled itself closed again. Here we go. See? It just widens that little opening at the top, and then out comes the little cilia, and then pop, everything pops open. That's pretty cool. And it sticks its little wiggly bits out there and tries to get food to get into its mouth. That's nice. Reasonably good view of it at 40x times 3. Let's see. That I don't have a scale bar for 40x times 3, but that's 40x, and so it's 3 times that. So that's 1/3 of a millimeter basically. How long do these things live? Most ciliates have pretty short lifespans. I mean, probably days, a month at most. They definitely don't live over the winter. So like, you know, if you find most of the ciliates, they don't live like a whole year, because they repopulate and recolonize lakes seasonally, basically. And you might get different ciliates living in the water depending on what time of year it is. So we looked at pond water from this early in the spring. I had some pond water from my koi pond on the light microscope in one of my evening streams, and I never saw anything like this in there. They might be waiting for, you know, they might take a while before the water gets to the right temperature for them to come out of their eggs, basically. So they might have special criteria before they'll hatch and then colonize the environment. Some of the organisms that we see, like the rotifers, are like the Dellard rotifers. They can handle like the water drying out, and they can actually be like exposed to open air, completely desiccate, and then come back to life when the water gets back on them. And so their lives are a little bit harder to track, generally speaking, to try to figure out how long they live. But everything that we've been looking at is just 2 drops of water or 3 drops of water from the beginning of the stream till now. I haven't changed the slides, and so it's a relatively tiny universe that they live in, and that's probably a long time for them. I mean, a couple of months is probably a really long time for them in the sense of the possibility of them being consumed, or yeah, this has all been like 2 drops of water. Keep in mind it's concentrated a little bit by the fact that I collected it from the side of the koi pond. So I scraped it with a bit of, I took one of the glass slides I have, and I scraped the side of the pond, and it's got like a kind of like a tarp that the whole koi pond is inside of, and the Cladophora grow on the tarp. So I just scraped a little bit of that tarp, basically. And we're super zoomed in. You can even look at some of these diatoms probably, because we're at the 400x scale. Is that the one we were just on, or is there another one? Oh, that's the one we were just on. Oh, here's one of these guys. Epistylus guys. We can maybe get a nice clean look at one of them as well. You can see them whirling their little cilia around. I can't remember whether it's Epistylus or what the other one is. But the cilia on these are on the edges, basically, and they spin them around, and then they're shaped sort of like little vases with a thread that's attached. So that's a pretty crazy view. So that's the scale bar is 100 microns, but we're 3 times magnification, so they're more like 30 microns. No? Is that right? 1, 2, 3. Yeah, I think it's like 30 microns or so. They're pretty small. 30 to 40 microns, yeah. Yeah, something like that. So, that's pretty nice. Oops. Let's see what else we can find. Oh, got a nice close-up of a rotifer. As long as we're right here. Oops, wrong button. There you go. Am I doing a thesis or a PhD project? I have news for you, VB Green. I am a full professor. I'm not a student. I haven't been a student for 2005 I got my PhD. So I've been a professor for 10 years, basically. I'm full professor now at Indiana State University. Is it just a research project? I'm doing about 15 research projects, if you include my work and the work that I'm on with my master's students and my undergrads and colleagues from different parts of the world. This is not actually part of my research. I study diatoms, which are the little things that these things eat. And I mostly use them for paleoecological reconstruction. So I have a paper that's coming out sometime soon in Global Change Biology, where we looked at the diatoms from June Lake with a student of Mike McLue's, and I have a species from June Lake as well that in California that I described a new species of diatoms from. I have, I just have projects all over the place, but most of my work is either taxonomy, diatom taxonomy, or diatom paleoecology. And then my students have projects in Iowa. I have a GIS student who has projects in Wisconsin. I have some projects in Indiana with some of my undergraduate students. I have projects in Idaho, Africa. I have a lot of projects in Africa going on right now. And I've worked in the Himalayas. I've worked in the Rocky Mountains, the Pantanal in Brazil. I think that's most of it. So I have projects all over the place. And Wyoming, Oops, here comes a nematode to mess everything up as well. So, but my research is largely focused on using diatoms to reconstruct past water quality, lake level changes, drought, across really long time scales, like thousands of years typically, and sometimes hundreds of thousands of years, and sometimes millions of years. So that diatom's crawling around. That's nice. Also, there's another rotifer over here that looks like it's trying to suck it up. Here it is. There's the diatom, it's crawling. And there's the rotifer. It's trying to suck it up in that vacuum cleaner shaped head of its got going on right there. Peter, thank you. Yeah, June Lake's pretty interesting. My colleague Mike has students working on Mono Lake, and I don't work on Mono Lake, but my master's student is working on Gull Lake, which is adjacent to June Lake. And we're also we're working on grants to do some research in the Mono Lake Basin as well, from all the lakes that are in that little Paleo Horseshoe Valley that's there. You're also visiting Mono Lake, yeah, good. It's I think it's a nice place to visit. It's a little bit more difficult as a place to do field work, but for my student Laura, who's just gotten back from doing field work out there, I think she really enjoyed it. So yeah, that guy is caught in that little Cenodesmus is caught in the current that's being generated right here. You see the Cenodesmus is just spinning. They can't move on their own. They're basically floaters, and I don't think they even have little like flagella or anything. I think they just float, and it's in for a long ride. I think it's going to be spinning there for a while, because even if it gets into the mouth area of the rotifer, it probably would reject that particle. It's making you dizzy, or is it making itself dizzy? You're planning to hit the natural springs in the area as well. Oh, that's cool. You're really going to be looking at a lot of really cool stuff. Studio, I know you said you were trying to reconstruct Husted's sort of travel path or something. Were you planning on like imaging the diatoms from all of that stuff? Are you planning on collecting sample? I'm curious, because if you're doing all that, you know, it might be nice to have samples from all of those spots for the SEM or the light microscope. You're collecting samples. So it's not for research, or is it for research? Whoops, it looks like the nematode came in and smacked our rotifer a little. From a biological function perspective, do rotifers do this as a hard-coded behavior? They're pretty simple creatures. So all of the ciliates, yeah, the Cenodesmus is pretty thankful that the rotifer has stopped, but I think that rotifer just moved a little bit. I think it's probably still around somewhere here, but they're not rooted. They have little toes. They can kind of crawl around using their toes. But you can really see those diatoms pretty clearly. That's nice. Yeah, the nematodes are they're all like that. They're all kind of rude. Oh, the Ehrenberg collection, okay. Oh, that's cool. It's for art projects, okay. There's the rotifer. You can see that one's crawling. They can kind of inch their way around, and they put that little spinning vacuum cleaner thing away when they're not using it to eat. So I guess your question, are they hard-coded to do that? I would think all microbes are basically hard-coded to do the things they do. That's just how they are. I don't think they make decisions in the simplest sense, like that's dangerous, that's not dangerous, that's where nutrients are, that's where nutrients aren't. But I think everything is basically instinctual, or mostly instinctual. Yeah. So, a lowly artist. Lowly artists are the best kind, aren't they? Oh, what's going on here? Giant nematode. Sneaking through our field of view. Like a dragon. Yeah, I do a lot of, as I'm sure you're aware, now studio, I do a lot of sci art using the SEM as my primary collection tool, and then colorizing the images that I collect. So I like to, pretty much every time I get on the SEM, take a few pictures that are just artsy pictures, in part to sort of showcase diatoms and how amazing they are. So, but I don't have any formal training in anything that I do other than diatom analysis. Most of my colorizing and everything else is just sort of instinctual decision making and tweaking things until it looks good to me. Oh, that's cool. I think this one is in that book. See the little bits in there that are spinning around, and then there's like an outer sort of bubble that they're all in. That's the good stuff, yeah. Well, it's the easy stuff, I guess. It's a lot harder to do art where you have to be trained and know what you're doing, and most of my photography as well is just instinctive decision making. I don't have any formal, I've never had like a photography class, but I have a lot of experience taking photos, so I feel like just gut trained like everything else except for the diatom stuff. What's this guy doing? Hello. He's just spinning around. So this one's shaped like a Good and Plenty candy with a bunch of cilia in rings around its body, and there's a set of rings at the bottom that it's using to spin around in circles. Jay Renzella, hello. I want to show you something, Jay. I hope you don't mind. I did this, hang on, I'm going to give you a shout out first. And then when I do, you'll see. Watch the screen. Watch the screen, John. Ah? You like? I stole some of your art and I put it in as a little emote that shows up. And it shows up anytime anybody says the word John. Try it. Just everybody in the channel try it. Let's see what happens. Just Jon. Just John, like that.
Daddy is cute. Daddy is strong.
What the heck just happened? Oh, we got a subscription. Well, now the Johns are going to get overwhelmed by bubbling diatoms and stuff. It should work though, if you just type John. It should like pop up or float or do something. But that's, oh, there it is. Yeah, it's down here. It's hiding. There's one. Bouncing. That's some of John's artwork. John's an artist who took some samples from SEM images and light microscope images from Pacific plankton and I, and then he's a woodcutter and a tattoo artist as well, and so he carves all of this stuff live on Twitch, and so he carved out the there was on the back side of that was an Actinopticus. Hang on, it's like this one, and then in front of it he had a little wiggly polychaete that he had used from Pacific planktons, and they were just combined together into that little colorful art wheel. And then after he does the prints, so he takes them, carves the wood, makes a print, and then he does like watercolor painting to paint them. So those are the real colors of the actual art. And then you can buy them. He has a little shop that he sells the stuff that he makes on Twitch, and he numbers them and everything. And then I think Pacific Plankton also then commissioned him to make some pretty cool stuff. But I thought that would be a nice little emote for you because it's almost circular. And then it will bounce across the screen, and then when I give you a shout out like this, like people go follow John, you know, go follow him, then it also like will pop up with them because your name's in there twice, right? So every time, if you just wrote John, it'll just make a whole bunch of them. So pretty cool. Sorry, I left like a blob of nothing on the screen while I was busy talking. Probably could have put it on something. Pretty cool. You really should go follow John. I'm serious about that though. You'll enjoy it, especially if you like the sort of chill atmosphere that I'm usually projecting in my streams. John's pretty chill, and we put one of his wood carving tools in the SEM once too for a special stream we did. Quick general question: for your cheaper microscopes for a 100x objective, does it have to be an oil objective? So D-Lurks. What I would say is you're probably not going to look at a lot at 1,000 times magnification. So for a 100x objective, you probably won't spend a lot of your time looking at stuff unless you're doing diatom analysis. But most 100x objectives. Whoa, what's happening? Ram's Reef and. Oh, Evil Lozzie and Scoopdom Cookies and J Runzella and Geyser Elf all got gifted subscriptions. That's amazing. Thank you for that, VB Green. That is very cool. Now you guys can spam the emotes as much as you like too. Just have to, you know, punch them in. That is very, very cool. So for people who are just here for the first time, checking out my wiggly things, the money from subscriptions and from cheers and from everything else that comes into Twitch, so Twitch takes a cut and then I get it, you know, like you pay $5 for a subscription, I get $2.50. I use all that money to pay for student research. I don't use any of it as like take-home money for the stream. I use it to either buy supplies for the stream, or I use it to like actually like give money for students to do research. So, somebody say Dell. Oh, it's delicate. I think it just punched that in when they did that. That's pretty funny. So thank you for supporting student research as well. So not only did you give some emotes to some cool people who are followers to the channel, but you also are supporting my graduate and undergraduate students who probably are very excited to know that there's somebody out there that's donating money to their projects or to help them present their research or whatever else that we can use the money for. So very cool. And I'm going to talk them into, when they use some of the money for travel or for like a presentation, to try to put the person's name, their Twitch name, into the acknowledgements, which I think would be pretty fun. So we'll put a list of Twitch subscribers or whatever in there in the acknowledgements for the posters. I think that would be pretty cool. Anyhow, so thanks for that. That's a bit of a long-winded thank you, but hello little wiggler. Are you streaming tonight, John? It's like a Wednesday. I don't remember if you streamed on Wednesdays. You're just here hanging out. There's a little wiggler. There's another little wiggler right there. Bunch of wiggly nematode worms. I think there was a bigger worm in here that we saw at the beginning of the stream. Hopefully it didn't slip out from underneath the... Let's see if I can get us there. I think it was here and then like this way. Somewhere in this mess. I saw a pretty big worm. A non-nematode worm. It wasn't doing a whole lot, it was just kind of hanging out in here. Oh, there's one of these guys. I saw one of these earlier, but it was eating. This one's not eating, it's just hanging out. It's got its mouth all closed. Not until tomorrow, okay. Just like to support the research. Okay, well, that's fine. Thank you for your subscription. I appreciate it. And so people get the fun little emotes they can use. So that's great. Try to do things a little bit differently here. Oops, I've already gone up to the top of this pile, thinking I was going downward because microscopes are upside down. Look at all those diatoms on stalks. You can really see the stalks that they're living on right there. They've got really long stalks. The pattern for diatoms to live on stalks, or for any periphyton to live on stalks, is it's about making more space. It's about space resources. Probably you've never thought about this, but if all these diatoms were living attached to that plant, and here's their attachment point, it would get really crowded. And their solution to this is to make these EPS, some polysaccharide, sort of long stalks, and then they can have a whole bunch of diatoms that are living like up here above that surface, where they have a little bit more room and can move around, where there's a little bit more water that moves past them, and potentially more light. And so that's part of their strategy, is like living on these long stalks that separate them from the substrate. It's the same way that you can get tiers of trees, like in a forest, where you have like the shade tolerant trees close to the ground, and then the farther you move up away from the surface. Yeah, it's just like plants. They're doing the same thing. And in fact, this model that I'm talking about is sort of designed on the idea of a forest. They call these periphyton forests. So periphyton just means, so phyton is like you know, things that have chloroplasts, and peri means they live around the outside edge. So they live attached to things around the margins of lakes. And they refer to this as a periphyton forest. It's like a little forest. And the diatoms that are there are, these ones are living at the, you know, the second story. Yeah. So they're functioning like a forest. And then there's other ones that are smaller that can't make these sort of structures, that basically are forced to live attached. And then there's ones that become truly filamentous, that are like long filamentous things to get way above the surface. But it's about space resources. So not like outer space resources, but like treating the surface that they're on as a limitation, right? So think about, like, there's nutrient resources that are limiting, and these organisms are largely living on sunlight, and so space becomes an important resource for them. Very cool. The nematodes are kind of neat when I'm not trying to look at something else. Oh, here's two of these guys, all just sitting right there on their chill little stalks. Spinning their little cilias. See this sort of fringe that goes around, and then you can see, even though they're tiny, even though they're like much smaller than those rotifers that we saw, the same thing's going on, where the little cilia are spinning and they're creating a current. It's just what ciliates do. It's part of their design so there's sort of like two main types of ciliates. There's these. They look like scrubbing bubbles. I actually have made that observation before. I don't think I've ever mentioned it, but I've thought of that before when I saw some. They look a lot like little scrubbing bubbles. If you're familiar with the commercial for those scrubbing bubbles that go like cartoon characters that go down the drain, they're almost identical to them. Really, they should file copyright infringements. But the ciliates usually have two different sort of techniques. They're using the ciliates like this to get around. So they spin them, and then basically they use it like a propeller to move in the water. Or they attach themselves and then they spin the cilia to bring stuff to them, because it's the only way they can move. And the funny thing is, the attachment points for these and for the rotifers and for many of the other things that live like this, that are filter feeders, they can become detached. And then you'll sometimes just see them go flying past, you know, like the field of view. And the rotifers are just using it to move. They literally look like a low-flying plane with its twin propellers. They have those like vacuum cleaner things spinning on the front. They let go with their foot, and then they just go like flying across the slides super fast. It's like a way for them to move very quickly. And so you seen earlier you saw the rotifers, they were kind of inching. The bdelloid rotifers were kind of inching, but they can also do this sort of propeller jet plane thing. And I've seen these guys also do that, where they become detached on the little stalks that they're on, and then they got their propellers going and they just go flying across the screen. I think it's on purpose. I think they're just moving to a different resource area, but I don't know for sure. It's hard for me to say for sure whether that's intentional or they just happen to get detached. So, okay, well, it's 12 midnight here, and I think I'm gonna stop streaming. We've seen a lot of really cool stuff, and what I'd like to do is go raid my friend Hannah, who is streaming right now, Hannah Rebecca Music. And Hannah is a musician on Twitch, and if